There is something happening right now that you can't stop, that you can't turn off, and that your body has been performing since long before you were born. Sound passes through you. You don't choose it. You don't consciously filter it. You don't invite it. It simply arrives, and your body transforms it into something the brain can recognize as music, as a word, as the crack of a branch breaking in the dark. What most people don't know is that this process doesn't happen in the ear. It happens despite the ear. The ear doesn't listen. The ear translates. And that difference changes everything. For centuries, humanity assumed that the ear was a passive receiver, a structure that captured sound from the environment and sent it to the brain in some vague and not fully understood way. The first anatomists who dissected the human ear in the 10th century were astonished by its internal complexity. But for a long time, they couldn't precisely explain why such a tiny structure, encased within one of the densest bones in the human body, was able to distinguish between the lowest note of a double bass and the almost imperceptible high note of a whistle. The answer took centuries to arrive, and when it did, it revealed a system of biological engineering so precise that it still inspires awe among the scientists who study it. To understand what the ear does, you first have to understand what sound is. Sound is not a thing. It is movement, a disturbance in air pressure, a series of compressions and expansions that propagate as a wave from the source. When someone speaks, their vocal cords vibrate, compressing the surrounding air, and these compressions travel outward in all directions, like the ripples created when a stone is dropped into water. The question the ear answers every moment is this: how do I convert these pressure variations into something the brain can interpret as information? The answer begins on the outside, in the most visible structure of the auditory system, the one we all recognize when we think of an ear. It is called the auricle, and its shape is not arbitrary. That irregular geometry, full of folds and curves that seem whimsical, is actually a refined acoustic capture and direction system honed over millions of years of evolution. The folds of the auricle subtly modify the incoming sound depending on its angle of origin. This difference in the acoustic signal is what allows the brain to calculate whether a sound is coming from above, below, in front, or behind, even when both ears receive virtually the same information. The auricle is, in essence, a passive analog equalizer that the body uses to locate sound sources in three-dimensional space. From the auricle, sound enters through the external auditory canal, a tunnel approximately 2 centimeters to 5 millimeters long that conducts sound waves to the first conversion structure, the eardrum. The eardrum is a thin membrane, only tenths of a millimeter thick, taut like the skin of a drum, that separates the outer ear from the middle ear. When pressure waves reach the eardrum, the membrane vibrates. It vibrates in a complex way, not with a simple, uniform movement, but with deformation patterns that encode the frequency and intensity of the sound striking it. A low-frequency sound displaces the eardrum slowly and with a greater amplitude. A high-frequency sound makes it vibrate quickly and with less amplitude. In that microscopic movement of a membrane not much larger than a small coin, translation begins. What lies beyond the eardrum is one of the smallest wonders of human anatomy. The middle ear contains the 3 smallest bones in the human body, so tiny that all 3 together would fit comfortably on a pinky fingernail. They are called the malleus, incus, and stapes, and their names accurately describe their shapes. The malleus is attached directly to the eardrum. The incus connects it to the stapes, and the stapes, the smallest of the 3 at just 3 millimeters long, transmits the vibration to the next stage of the system. These 3 ossicles form a mechanical chain that amplifies the movement of the eardrum. But they don't simply amplify it, they transform it. The area of the eardrum is considerably larger than the area of the base of the stapes, which concentrates the force of the vibration in a much smaller space. This significantly increases the pressure, enough to overcome a fundamental physical problem: the resistance of the inner ear fluid to being disturbed by signals arriving from the air. Because what lies beyond the ossicular chain is not more air, but a fluid. And that is precisely what necessitates all the mechanical machinery of the middle ear. When a sound wave travels through air and attempts to penetrate a liquid directly, most of the energy is reflected at the interface. The middle ear solves this energy transfer problem by acting as an acoustic impedance transformer, a concept from physics that, in practical terms, means the ossicles adjust the signal so that it reaches the fluid in the inner ear with enough force to effectively disturb it. The inner ear, also called the labyrinth due to its anatomical complexity, contains two distinct but intertwined systems. One controls balance, the other controls hearing. The structure responsible for hearing is called the cochlea, and if you could remove it from the temporal bone that houses it and unfold it, you would see that it is a tube coiled upon itself in a spiral shape with approximately 2.5 turns, with a total length of about 35 millimeters. Inside this tube, separated into longitudinal compartments by membranes, circulate two fluids with distinct chemical compositions, perilymph and endolymph. The membrane that separates them in the central region of the cochlear duct is called the basilar membrane, and it is here that something extraordinary occurs. The basilar membrane is not uniform. It is wide and flexible at the apical end of the cochlea and narrow and rigid at the basal end. This difference in mechanical properties along its length causes different regions of the membrane to respond preferentially to different frequencies. Low-frequency sounds generate maximum vibration near the apex of the cochlea. High-frequency sounds vibrate primarily at the base. When the stapes transmits the vibration to the cochlear fluid, a wave is generated that travels along the basilar membrane and reaches its maximum amplitude at the point corresponding to the frequency of the original sound. The cochlea, in a way, breaks down sound into its constituent frequencies, just as a prism breaks down white light into its colors. But the cochlea does this in real time, with a speed and precision that no artificial device has yet fully matched. The organ of Corti, the structure many scientists consider the true auditory transducer, the place where mechanical vibration is converted into an electrical signal, sits on the basilar membrane. The organ of Corti contains approximately 15,000 hair cells distributed along the length of the basilar membrane. These cells are named for the projections that emerge from their apical surface. Stereocilia, tiny hair-like structures that are grouped in a V or W shape. When the basilar membrane vibrates, the stereocilia bend, and it is at that moment, in that tiny movement of structures measured in micrometers, that the conversion which makes hearing possible occurs. The stereocilia are connected to each other at their tips by protein structures called tip links, molecular filaments that link the top of one stereocilium to the side of the next taller one. When the stereocilia bend in the correct direction, the tip links tighten and open ion channels in the cell membrane. Through these channels, potassium and calcium ions flow into the cell, following concentration gradients, and this ion flow generates a change in the cell's electrical potential. This electrical change triggers the release of neurotransmitters at the base of the hair cell, which activate the auditory nerve fibers. Mechanical vibration has been converted into a nerve impulse. Sound has been transformed into electrical language. There are two types of hair cells worth distinguishing. Inner hair cells, approximately 3,500 in total, are primarily responsible for transmitting auditory information to the brain. Outer hair cells, which are about 4 times more numerous, do something entirely different and, for a long time, unexpected. They actively amplify and fine-tune the mechanical response of the basilar membrane. These cells are able to change their length in response to changes in their electrical potential thanks to a protein called prestin, which acts as a molecular motor. Through this mechanism, the outer hair cells adjust the cochlear mechanics in real time, improving the sensitivity and frequency resolution of the system. This active amplification is the reason why the human ear can detect incredibly low-intensity sounds and why it can distinguish two frequencies that differ by less than 1%. The electrical signal generated in the hair cells travels through the cochlear nerve, which is part of the 8th cranial nerve, also known as the vestibulocochlear nerve. This nerve conducts impulses to the brainstem, where the information is distributed through multiple nuclei and ascending pathways to the primary auditory cortex, located in the temporal lobe of the brain. What occurs along this pathway is of a complexity that science still does not fully understand. Auditory information does not reach the brain as a simple signal. It arrives encoded in temporal and spatial activation patterns of thousands of nerve fibers. And the central nervous system extracts information from these patterns about the frequency, intensity, timbre, spatial location, and duration of each sound. One aspect of hearing deserves special consideration: its astonishing sensitivity. The human threshold of hearing, the minimum sound pressure level the ear can detect under ideal conditions, corresponds to displacements of the basilar membrane on the order of tenths of a nanometer, figures comparable to the diameter of hydrogen atoms. At the threshold of perception, the stereocilia bend at such small angles that they are difficult to visualize, yet the system detects them. This sensitivity comes at a cost. The same exquisite machinery that allows us to hear a whisper across a silent room can be permanently damaged by prolonged exposure to loud sounds. Unlike other cells in the body, hair cells do not regenerate in adult mammals. When they die, they are gone for good. This is why noise-induced hearing loss is irreversible and is also an active field of research. Scientists in various laboratories are exploring whether it is possible to induce hair cell regeneration using growth factors or gene therapy. Preliminary results in animal models are promising, but translating that knowledge into clinical treatments for humans is a path that is still far from complete. There is another dimension of hearing that is rarely mentioned when discussing the ear: the inner ear. The brain is not a passive receiver of auditory information. The central nervous system sends active signals back to the cochlea via the olivocochlear system, modulating the sensitivity of the outer hair cells according to the context. When you're in a noisy room trying to overhear a conversation, or when you anticipate a loud sound, the brain actively adjusts the inner ear's game to optimize perception in that specific context. Hearing, in that sense, is not just a flow of information from the world to the brain. It's a continuous dialogue between the brain and the ear. Another frequently misunderstood concept is that of silence. The human brain does not experience silence as the absence of a signal. When the environment is sufficiently quiet, the auditory cells themselves generate spontaneous activity that the brain perceives as background noise. This explains why, in an anechoic chamber, a room designed to absorb virtually all sound, many people begin to hear sounds coming from inside their own bodies: their heartbeat, blood flow, the movement of air in their lungs. In the absence of external stimuli, the auditory system turns inward. Research on central auditory processing continues to reveal unexpected layers of complexity. The auditory cortex, for example, is not simply a map of frequencies. Functional neuroimaging studies suggest that different regions of the cortex process different aspects of sound. The identity of the sound source appears to be processed along a pathway that runs to the anterior regions of the temporal lobe, while the spatial localization of sound involves more posterior regions. Recent evidence also indicates that the perception of speech and music activates distinct processing patterns from very early stages of the auditory pathway. Although there is still an active debate about the precise mechanisms, what is clear is that the human ear is much more than a biological microphone. It is a multi-stage energy conversion system, each stage specialized in solving a specific physical problem: capture and direction in the auricle, impedance matching in the middle ear, frequency analysis in the cochlea, and mechano-electrical transduction in the organ of Corti. And all of this occurs in a structure whose most delicate part fits inside a bone the size of a cherry, housed within the skull to protect it from impacts and extreme temperatures. When we stop to think about what happens every time sound reaches our ears, something changes. The most ordinary conversation, the most commonplace music, the sound of rain against a window, cease to be mere experiences and become the visible result of a chain of physical, mechanical, fluid, and electrical transformations that the body executes in milliseconds, without our intervention, without our conscious decision, with a fidelity and precision that even the most advanced technology is only beginning to approach. The ear doesn't hear the world, it translates it. And in that silent, invisible, incessant translation lies one of the most refined architectures that evolution has produced in the entire history of life on this planet. Every time you hear something, your body executes in milliseconds a chain of transformations that took science centuries to decipher. The ear is not a simple organ. It's an extraordinarily precise system, built over millions of years of evolution, capable of detecting movements smaller than an atom and translating them into experience. That's happening to you right now, without your conscious awareness. If this journey through the human ear has taught you something new, please consider liking this post. It's the simplest way to tell the world that this kind of knowledge matters. And if you want to continue exploring the hidden architecture of the human body, subscribe to Anatomy Revealed. There's so much more to discover.